Hello and welcome to my YouTube channel. Today's topic is DNA polymerase types and their functions. Prokaryotic DNA polymerases. So there are 5 DNA polymerases identified in E. coli. All the DNA polymerases differ in structure, function and rate of polymerization and their processivity. DNA polymerase structure. DNA polymerases are a group of enzymes that carry out DNA replication or polymerization, and not only do they polymerize but they also do proofreading and thereby they prevent mutations. So the structure of most of the DNA polymerases resembles a hand which is holding active sites. So this is the hand, and now we'll look at the protein DNA polymerase. This is the DNA polymerase which has primarily 2 active sites at least. So this is the polymerizing site and this is known as the polymerase site, and the lower part of the hand which has the exonuclease activity. So the polymerizing activity is from 5 prime to 3 prime, whereas the exonuclease activity is from 3 prime to 5 prime. Now this is a comparative table of 5 different types of prokaryotic polymerases. We will take a look at that. So we will assess them by the type, the gene coded by them, the 5 prime to 3 prime polymerase activity. This 5 prime to 3 prime polymerase activity is exhibited by all the polymerases, exonuclease activity and different subunits present in each polymerase. DNA polymerase 1 is coded by POL A gene. It has the 5 prime to 3 prime polymerase activity. Its main function is to carry out DNA repair. It removes and replaces RNA primers, fills the gap after removal of RNA primer. Exonuclease activity exhibited by DNA polymerases 1 is of 2 types. It has 5 prime to 3 prime as well as 3 prime to 5 prime exonuclease activity. Now this is a unique feature of the DNA polymerase 1, as you will see later on that it is the only DNA polymerase that has activity, exonuclease activity in both 5 prime to 3 prime and 3 prime to 5 prime direction. It is made up of a single polypeptide chain. DNA polymerase 2 is coded by POL B gene. It carries out DNA repair, starts replication after damaged DNA halt synthesis. It has exonuclease activity in 3 prime to 5 prime direction, and it is made up of 7 subunits. The DNA polymerase 3 has 2 subunits that act as sliding clamp. These are the 2 beta subunits. We will take a look at the structure a little later. It is coded by POL C gene. Its major function is elongation of DNA during replication. It also carries out proofreading and editing. It has only 3 prime to 5 prime exonuclease activity. It is made up of 13 subunits. DNA polymerase 4 is coded by DIN B gene, and the DNA repair during SOS response is its major function. Exonuclease activity is not present in polymerase 4, and the subunits, it's made up of a single polypeptide chain. The DNA polymerase 5 is coded by the UMUDC gene. Its major function is DNA repair, translesion DNA synthesis. It does not have any exonuclease activity, and it is a trimer made up of UMUC monomer plus UMUD dimer. It is also represented as UMUD2C. Now, DNA polymerase 3 is the main replicating enzyme in prokaryotes because it has this clamp activity, which will be explained soon. DNA polymerase 2, 4 and 5 carry out replication even when there is DNA damage, and this kind of replication is known as translesion DNA synthesis or SOS response. Let us take a look at the DNA polymerase structure, and you can see here that it has 5' to 3' polymerase domain, and it also has 3' to 5' as well as 5' to 3' exonuclease domain. So basically all these three domains together make a DNA polymerase 1 holoenzyme. This is the complete enzyme. If this enzyme undergoes proteolysis, which is possible in vitro in a laboratory, then there are two fragments that are formed. One is the smaller fragment having 5' to 3' exonuclease domain, and the second one is the larger fragment which has the polymerase domain as well as the 3' to 5' exonuclease domain. This is known as the Klinau fragment, and it is the larger fragment of the two of DNA polymerase 1 enzyme, and this Klinau fragment is useful when polymerization activity is needed in vitro conditions. For example, in filling of 5' overhangs, in sequencing DNA, and in synthesis of double-stranded DNA, and synthesis of the active site of the enzyme has two parts. The first is the 5' to 3' polymerase activity at the polymerase site. Second is the 3' to 5' exonuclease site, and in some polymerases there is 5' to 3' exonuclease site. Example of such DNA polymerase is DNA polymerase 1. So this is the typical DNA polymerase enzyme with two active sites, that is the 5' to 3' polymerase activity and the 3' to 5' exonuclease activity. These are the two commonly found active sites in a DNA polymerase. Now in DNA polymerase 1 there is a third site, that is the 5' to 3' exonuclease site. So the DNA polymerase 1 has the polymerase site, the 3' to 5' exonuclease site, and the 5' to 3' exonuclease site. So it has three active sites. Now let us look at the difference between gaps and nicks. As you know that DNA polymerase is capable of filling up the gaps. So are gaps and nicks exchangeable terms? Or are they synonyms, or do they have two different meanings? So whenever a replication process is going on, as you can see in this strand, a small gap is here. The bond is not present over here. However, the nucleotides in both the strands are present. But the bond between the hydroxyl group and the phosphate group is not formed. So in this case, where complementary nucleotides in both strands are present, but at certain places the hydroxyl group and the phosphate bond have not been able to form the phosphodiester bond, that is known as the nick. So this is the nick, and this is another example of a DNA molecule in which you can see that in one strand all the nucleotides are there, whereas on the other strand at one place the nucleotide is missing. So when the nucleotide is missing, it is known as a gap, and the nicks are sealed by ligases, whereas the gaps are filled by polymerase enzyme. So this is the difference between gaps and nicks. For the ligase activity, ATP is required. A major use of the DNA polymerase 1 is in the nick translation process for radio labeling. Let us take a look at the DNA polymerase structure. The DNA polymerase 3 we will be discussing here. Structure appears like a man kneeling down with his arms open and the fist on top. And you can see this is the structure. It's similar to this. Now we will individually see each part of the polymerase. It has several subunits, and let us see what these subunits do. So the arms are made up of theta, epsilon and alpha, and they are exist as dimers, and they form the core of the enzyme. So this is the core of the enzyme which carries out the polymerization. The second part is composed of the tau region which forms the body, and it keeps the dimer intact and together. The third part of the enzyme is composed of the clamp loader, and this clamp loader is made up of two gamma, chi, psi, delta prime and delta. So these are the different parts of the clamp loader. The function of the clamp loader is to load the clamp. Now this beta subunit is the clamp which is separate from the enzyme, and the clamp loader loads it to the arms or to the core of the enzyme. So this is how the clamp will be loaded, and then this whole thing will become a holoenzyme. Now the function of the tau is to keep the whole enzyme together, and the function of clamp loader is to load the beta subunit to the core enzyme. So let us see. So let us take a quick look at the polymerase 3 holoenzyme and the functions of different functional subunits. There are four functional subunits. The first one is the core enzyme, which elongates the polynucleotide chain and proofreads. This is the core enzyme area. Then it is composed of the gamma complex. This is the gamma complex, which is known as the clamp loader, and it also consists of two beta subunits which are the sliding clamp. They are also known as the processivity factor, which increases the efficiency of polymerase while replication, and toe subunits help the dimer to stay intact while the leading strand and lagging strand synthesis is going on. This is the replicating DNA molecule. This is the leading strand. This is the lagging strand. This is a single DNA polymerase 3 enzyme which is carrying out replication in both the leading as well as the lagging strand. So since the single enzyme is doing this job, there is a loop formed in the lagging strand, and this is known as the DNA loop. Now the DNA polymerase has two main functions. One is polymerization. The polymerization property is decided by the polymerization rate, which is defined as number of nucleotides added per unit time. And the second function is processivity, which will be discussed later on in detail. It is defined as the number of nucleotides added by the polymerase enzyme before it falls off. Now this processivity is increased by the beta clamp, and if this beta clamp does not exist, then the processivity of the DNA polymerase drops down. Now we'll come to the eukaryotic DNA polymerases. There are at least 15 known DNA polymerases identified in human beings. The main 5 DNA polymerases are alpha, beta, gamma, delta and epsilon. The polymerase gamma is located in the mitochondria, and the remaining polymerases, that is alpha, beta, delta and epsilon, are found in the nucleus. Let us take a look at the eukaryotic DNA polymerases, their types and the functions. The DNA polymerases in eukaryotes have various types, but here we will discuss only the 5 important types. So, the DNA polymerase alpha has a primase function. It initiates DNA synthesis on both leading strand and lagging strand by synthesizing a RNA DNA hybrid primer. It does not have any exonuclease activity, and has 4 tetrameric subunits. The DNA polymerase beta main function is DNA repair. It does not have any exonuclease activity, and it is made up of a single polypeptide chain. DNA polymerase gamma is the main enzyme that replicates only mitochondrial DNA, and it is present in the mitochondria. It has 3' to 5' exonuclease activity, and it is a dimeric protein, so it has 2 subunits. The DNA polymerase delta is the enzyme for replication in lagging strand, and it has 3' to 5' exonuclease activity. It is tetrameric in nature. The DNA polymerase epsilon is the enzyme for replication in leading strand, and it also repairs DNA. It has 3' to 5' exonuclease activity. It is tetrameric. The DNA polymerase epsilon is the main replicating polymerase in eukaryotes, whereas DNA polymerase delta has PCNA clamp, so it has high processivity. What is processivity? We will discuss shortly. Now let us look at the difference between a mammalian and E. coli DNA polymerase. So here we will be comparing with E. coli and mammalian polymerase. The DNA polymerase 1 in the prokaryotes does the gap filling work, the polymerase 2 does repair work, and the DNA G does the primer work. It is known as the primase. It is a separate enzyme, and this synthesis of primer is carried out by the DNA polymerase alpha in eukaryotes or in mammals. The DNA polymerase beta does DNA repair work, the gamma does mitochondrial DNA synthesis, the delta polymerase carries out lagging strand synthesis, and the epsilon polymerase carries out leading strand synthesis. So the DNA polymerase 3 of E. coli and DNA polymerase epsilon of the eukaryotes are comparable because both are the highly processive enzymes carrying out the synthesis of leading strand. Now, what is processivity? We will look at later. Processivity. The processivity is defined as the ability of DNA polymerase to carry out continuous DNA synthesis on a template strand without frequent dissociation or slipping off from the strand. The DNA clamp is also known as a sliding clamp, and it is a processivity promoting factor in DNA replication. So in E. coli, polymerase 3 adds up to 250 to 1000 nucleotides per second in the presence of a clamp. Its processivity is around 5 lakh, and in eukaryotes it's the DNA polymerase delta that adds up to 150 nucleotides per second. Clamp and clamp loaders. Clamp and clamp loaders are proteins that bind to DNA polymerase and prevent it from dissociating from the template DNA strand. This helps the DNA polymerase complex to stay on the DNA through the entire cycle of replication. A multi-subunit complex functions as the clamp loader, which we have seen earlier, and it loads the clamp onto the DNA. In case of prokaryotes, it was the beta clamp, which was being loaded by the clamp loader unit of the enzyme. We have already discussed about the DNA clamp protein in prokaryotes earlier. Let us look at the DNA clamp protein in eukaryotes. In eukaryotic cell, there is a multi-subunit protein called the replication factor C, RFC, which is the clamp loader, and the proliferating cell nuclear antigen or the PCNA, which is the sliding clamp. So the clamp loader is replication factor C and the clamp is proliferating cell nuclear antigen for eukaryotes. PCNA is a DNA clamp that acts as a processivity factor for DNA polymerase delta in eukaryotic cells, and it is essential for replication. If you remember, the DNA clamp in prokaryotes is the beta homodimeric clamp. Now you can take a look at this. This is the clamp. This is the clamp loader. It requires energy to open the clamp, and then the DNA is this clamp loader enables the clamp to fix to the DNA with the help of ATP. Once the clamp is fixed to the DNA, the clamp loader will dissociate and the polymerase will bind to the DNA, and the polymerization or replication will start. So this is the beta dimer of prokaryotes, which is the clamp, and in eukaryotes it is the PCNA. Beta dimer is a dimeric protein and it is found in prokaryotes. It is the clamp protein. PCNA is a trimeric protein, which is a clamp protein of the eukaryotes. Thank you for watching. Like, comment and share and subscribe to my channel.